Welcome to our educational webinar entitled Understanding Pancreatic Development and Diabetes Using Patient-Specific iPS Cells. My name is Jenna Mocha and I am a Product Marketing Manager at Stem Cell Technologies. I will be your moderator for this webinar today. It is now my pleasure to introduce you to our two speakers today. Dr. Ray Dunn is a principal investigator at A-Star Institute for Medical Biology in Singapore and holds an adjunct assistant professor position at the Lee Kong Chian School of Medicine at Nanyang Technical University and the NTU School of Biological Sciences. Dr. Jamie Trott is a postdoctoral researcher in Dr. Dunn's laboratory. Today, Ray and Jamie will describe some of their research to develop in vitro models of pancreatic development and diabetic cell failure. Let me now hand you over to Ray to begin the presentation.
Thank you, Jenna, for the introduction, and I'm the kickoff speaker, Ray Dunn. My lab has had a long-standing interest in modeling human pancreatic development in vitro, and typically this is accomplished by the directed differentiation of human pluripotent stem cells, either human embryonic stem cells or human induced pluripotent stem cells, and through the provision of growth factors, small molecules, and medium formulations, we can directly differentiate these cells into obligate intermediate stages, the first of which is the definitive endoderm, the parental germ layer of the pancreas, followed by the acquisition of positional identity and pancreatic fate. And finally, these pancreatic progenitor cells go through stages of differentiation that make them into endocrine progenitors, which ultimately can give rise to these highly specialized cells called beta cells, which sense glucose in real time and secrete insulin. Ultimately, the therapeutic goal is to produce these cells in sufficient numbers in a clinically compliant fashion to treat type 1 diabetic patients. Along this journey from pluripotent stem cells to the terminally differentiated beta cell, one also has the opportunity to study at high resolution and to obtain a very granulated understanding of these intermediate stages that pluripotent stem cells must transit through as they journey to the beta cell. And by far one of the most interesting transcription factors, and maybe the most famous one involved in this process, is PDX1. PDX1 labels the developing mammalian pancreas, and in the it encodes a homeobox protein, transcription factor, and you can see in these images from the mouse embryo going from left to right that we have immunofluorescence images of the developing pancreas from day 9 of gestation to day 11.5. The PDX1 protein beautifully labels the early dorsal pancreatic bud and the ventral pancreatic bud. These buds proliferate and expand and undergo a specialized form of branching morphogenesis called tubulogenesis to give rise to the early pancreatic onlogon. This expression is evolutionarily conserved in humans, and a couple of years ago, Neil Hanley's lab in Manchester reported that PDX1 indeed labels the ventral pancreatic and dorsal pancreatic regions of the developing endoderm that will eventually give rise to the human pancreas. PDX1 is not only this really beautiful and conspicuous marker of the developing pancreas, it is also genetically required. And now many years ago, PDX1 was inactivated by a couple of groups, one of which is Chris Wright's group at Vanderbilt University. And the top panels show the loss of function pups that are born, and you can see that the mutant pup looks a little smaller than the wild type and has milk in its stomach, so it was able to suckle with its mother. 6 days later, though, this pup has gone through a severe wasting syndrome and is in fact extremely hyperglycemic. And the reason for that is if you do a necropsy on these pups, you'll find that a wild-type pup has a pancreas shown here, but the mutant is entirely lacking the pancreas. So a single mutation in a single gene leads to a very dramatic focal pathology of pancreatic agenesis. And in 1997, the first inactivating mutation of PDX1 was identified in humans, and in this case, a child was born also lacking a pancreas based on sonogram at birth, and she required both the provision of insulin and exocrine hormones in order to survive beyond the postnatal period. In addition, if you go to OMIM and then look for other alleles of PDX1 that have been identified you'll find that it is indeed a rare mutation in humans. And in this list that you see in the screen, there are a few mutations that have been identified that are associated with adult onset diabetes. And this is because PDX1 is also expressed in the insulin-secreting beta cells in the human pancreas. So loss of PDX1 also impacts islet homeostasis. And the role of PDX1 both in the embryo and in the adult really intrigued my lab and I, because PDX1, although quite famous, its transcriptional targets had not yet been really fully identified or fully understood. So we chose to grapple with this question by performing directed differentiation in vitro with human embryonic stem cells into early pancreatic progenitors. And I want to emphasize here that we're looking at cells that first express PDX1, like those in the early pancreatic bud that I showed you in the mouse and human images earlier in my talk. So through protocols that we developed in-house over the last decade or so, if after roughly 17 days of differentiation, you can see that the cultures have changed dramatically from their colony phenotype to these little mountain ranges and honeycomb pattern, and it's within these thickened ridges that if you do immunofluorescence, you can find very abundant numbers of PDX1 positive pancreatic progenitors. These cells also express another transcription factor called SOX9, and based on extensive work in the mouse embryo, it is thought that these double positive SOX9 PDX1 cells are indeed the proliferating progenitor pool in the developing pancreas. And just to point out what stage this would be in humans, this is around 4 to 5 weeks of gestation in humans. So using some high affinity antibodies provided to us by Chris Wright, we then took cultures and performed ChIP-seq, which is probably quite familiar to the listening audience. And we were quite happy to observe that in the motif analysis, the most enriched motif is the PDX1 binding motif in our ChIP-seq that was done on day 17 of differentiation. We then took this data set and aligned it with companion comparative microarrays that we had performed over time. This is day 0, day 10, and day 17, done in quadruplicate, and we simply wanted to see which genes were bound by PDX1 whose expression was also increased on day 17 of differentiation. And we found, looking at this time only within 20 kb of the transcriptional start site, that roughly 350 genes were upregulated. Among these were new and novel genes, which have also, which of course have captivated our imagination, but we began to focus on one that was also quite well known, and that gene is RFX6. RFX6 stands for regulatory factor X6. There are 7 members of this winged helix transcription factor subfamily. RFX6 is beautifully expressed in the pancreatic endoderm, much like PDX1. And you can see here in these immunofluorescence images that RFX6 labels the dorsal pancreatic bud, as well as other endodermal cells anterior and posterior to the developing pancreatic bud. Here again is the companion immunofluorescence for PDX1 and co-localization of RFX6 and PDX1 in the early pancreatic onlogin. In the right-hand panel is just another way of visualizing RFX6 expression, and this is using a LAC-Z reporter system, and in this case you can see that RFX6 labels the lung, the stomach, the dorsal pancreas, and the gallbladder, but is conspicuously absent from the liver. Knockout studies were also performed in this paper published now many years ago by Smith et al. In Nature, and the RFX6 mutant pups, like PDX1 null mutants, are born. They die within 2 days of birth. They're quite a bit smaller, and they have this very bloated abdomen, and also they have a very and they also have a hypoplastic pancreas. The authors of this study went on to investigate at high resolution what actually is happening with the pancreas during development. And what they show in these panels is that on day 17.5, wild-type embryos show quite extensive expression of this secretory protein chromogranin A in endocrine cells within the developing pancreas. And these chromogranin A cells also contain hormones, here insulin, glucagon, and somatostatin. In the mutants, it was observed that none of the chromogranin A positive cells, the endocrine cells, contain hormones. So it was then hypothesized that the the RFX6 actually is downstream of this transcription factor called NGN3, which is a basic helix-loop-helix transcription factor. And it had been shown in several genetic studies in the mouse that NGN3 is absolutely required for endocrine differentiation. In these mutants, chromogranin A shows you that you have endocrine cells, but failure to activate the hormone program. So this was independently, just to point out, confirmed 2 years ago by another study showing that conditional inactivation of RFX6 in the mouse results in the same phenotype. In humans, RFX6 mutations result in a syndrome called Mitchell-Riley syndrome. Including the allele that I'm about to introduce to you, there are 9 alleles in OMIM that have been associated with Mitchell-Riley syndrome to date. These patients have a phenotype that mirrors the mouse phenotype, including hypoplastic pancreas, neonatal diabetes, duodenal atresia, and these patients usually die within the first year of life. Given that we had identified RFX6 as a downstream target of PDX1, we were quite excited by the fortuitous finding of our collaborator Bruno Reversade, whose lab is next door to us, where he had identified a Syrian patient with a homozygous nonsense mutation in human RFX6. And this young girl died within just a few days of birth with a hypoplastic pancreas, excuse me. We were able to obtain fibroblasts from this young girl and to derive using Sendai virus human iPS cells containing this homozygous nonsense mutation. So we were very interested, given that we had this reagent, to model what are the consequences of RFX6 loss during human pancreatic differentiation in vitro. From the work that I explained to you about the mouse, we hypothesized that the RFX6 would play a role downstream in the endocrine program. We reasoned that RFX6 in vitro in humans would function in those chromogranin positive cells before the acquisition of the hormone program. And so we thought that it would be necessary to implement a differentiation program which efficiently allowed us to recover endocrine precursor cells. Our lab, however, apologies, it doesn't let me, however, had been principally focused on these early pancreatic progenitors, those cells which emerge from the dorsal and ventral pancreatic bud during early stages of differentiation. So we had to entertain other differentiation platforms which would allow us to then directly differentiate the RFX6 deficient cells into the endocrine lineage. So to do this, we of course were drawn to a trio of papers that received much attention in the last year and a half, from the Melton Lab at Harvard, the Kiefer Lab at the University of British Columbia, and the Hebrock Group at UCSF in California. And these protocols showed significant improvement over previous differentiation platforms for the production in vitro of glucose-sensing, insulin-secreting beta cells. In particular, we began to focus on the 7-step protocol developed by Rosania et al. And published in Nature Biotechnology. Fortunately for us, a component of this protocol, which I mentioned was 7 stages, was under development as a commercial kit by Stem Cell Technologies. And this is outlined at the bottom of my screen. So we thought that one way of implementing the Kiefer protocol in our lab was to use this batch-tested kit as a way to differentiate both our human embryonic stem cells and our human iPS cells alongside to determine at which stage RFX6 was critical for further endocrine differentiation in humans. So we first validated this, and the cell line that we chose to validate is the very popularly utilized human embryonic stem cell line H9. And you can see that over time that the pancreatic markers SOX9 and PDX1 begin to upregulate their expression around day 7. And here on the right-hand side of the screen, you can see that RFX6 expression activation coincides quite nicely with the onset of PDX1. 2 other very important pancreatic markers are NKX6.1 and NGN3, and they too show robust upregulation beginning around day 7 of differentiation and increasing to the end of the protocol, which is day 15. We then wanted to evaluate whether our human iPS lines derived from our RFX6 deficient patient would differentiate equally as well. And so we did this systematically. So we then assayed at day 4, which is the stage of definitive endoderm, by comparing our wild-type H9 cells and our human iPS. And you can see in this immunofluorescence image just with one clone of the iPS that we upregulate classic markers of the definitive endoderm lineage, GATA6 and SOX17, where almost every cell expresses these two transcription factors. If you then quantitate this by FACS, you can see that the number of SOX17 CXCR4 positive cells, which indicate definitive endoderm, is roughly the same between the H9 cells as well as our two clones from our RFX6 mutant patient. I do want to point out that the RFX6 iPS clones you can see from the immunofluorescence as well as the cell counts and the FACS are fewer than the wild type, although they were seeded at different densities at the very beginning of differentiation. And this may be something that we will follow up on in later, but it could be that there is a proliferative issue or possibly apoptosis, but we're still not unclear why we lose some of the cells during the early differentiation. However, if you move on to the primitive gut tube stage, you can see that two classic markers of this stage, FOXA2 and HNF6, are equally expressed between the wild-type H9 cells and the RFX6 iPS, and this is also observed by doing immunofluorescence. Moving to day 10, however, things become a little bit more surprising. We expected, based on the studies in the mouse, that our iPS cells would differentiate to this stage and then progress further to day 15. That was not what we observed. In fact, the iPS showed collapse of SOX9 expression, collapse of PDX1 expression, and significantly diminished HNF6 expression. And this is also observed by doing immunofluorescence. So this suggested, using our two iPS clones from our RFX6 mutant patient, that RFX6 might play an earlier role in pancreatic lineage differentiation than previously suspected from work in the mouse. If we take the protocol to the very end on day 15, you can see by doing FACS sorting for NKX6.1 and PDX1 positive cells that our wild-type cells, wild-type H9 cells, we recover roughly 94%, whereas in the iPS clone, number 1 and 2, we have very few PDX1, NKX6.1 double positive cells. Again, emphasizing that RFX6 has an earlier role in pancreatic development than previously thought in humans. So I showed this slide earlier to emphasize that our idea based on work in the mouse was that RFX6 functions at this critical stage when cells are becoming mature endocrine cells or hormone-containing cells. But in fact, our iPS data show that the RFX6, at least in the human system, operates at an earlier stage. RFX6, because it's expressed at the same time as PDX1, could potentially regulate PDX1, and I've shown you by ChIP-seq that PDX1 is bound to RFX6 in human pancreatic progenitor cells. So we believe that this regulatory relationship in humans is slightly different than what has been previously reported for the mouse. So what is next for us? We have some additional controls to perform, one of which we have obtained recently, heterozygous iPS line for this RFX6 mutation from the father of the child that I mentioned earlier. And we're using CRISPR-Cas9, which I'm sure many of you are using out there in the audience, to inactivate activate RFX6 and H9 cells, and this will provide us with a an ideal isogenic control to mitigate any criticism out there that the phenotypes that we have observed are iPS dependent and not due to the genetic lesion in the cells. Once we've performed these controls, our goal is to perform comparative RNA-seq to identify those genes whose expression is absolutely dependent upon RFX6 and those genes which are bound by RFX6 using a ChIP-seq. And so in the first part of this talk, I've discussed a known mutation, RFX6, which was now first published about maybe 5-10 years ago, and the power of using reproducible differentiation platforms to interrogate the role of known transcription factors during pancreatic development. In the next part of the talk, I'm going to pass the baton to Jamie Trott, my postdoc, who's going to move from discussing what we know about known transcription factors to using patient-derived iPS cells to identify novel transcription factors or other genes responsible for beta cell maturation in vitro using patient-derived iPS lines. Jamie?
Okay, so thank you very much, Ray, for the introduction. As Ray just mentioned, my project in his lab is to try and identify novel genes and pathways that are specifically involved in the development and the function of human beta cells. So to do this, I'm studying diabetic families in which inheritance of a single unknown mutation leads to beta cell failure and diabetes. So how do I do this? So on the left here you can see a schematic outlining my project. So the first step is to identify these families. Then once I've identified them, I undertake a thorough genetic analysis in order to identify the pathogenic mutation and the target gene that it affects. So once I've done this, I want to know what's the effect of this mutation. So to do that, I take iPS from the diabetic patients and differentiate them firstly into pancreatic progenitors and then subsequently into beta cells in order to look at the effects of these mutations on the physiology of the diabetic beta cells. And then finally, in the future, I'm looking to use CRISPR genome editing to correct those mutations in order to show definitively that a particular mutation that affects the activity of a particular target gene leads to particular physiological defects that cause diabetes. So on the right here you can see a pedigree diagram showing one of about 20 Middle Eastern families we've identified that appear to exhibit an inherited form of beta cell failure. So our criteria for identifying these families are fourfold. So firstly, they should have an exceptionally high disease incidence. So in this case you can see that 1, 2, 3 out of the 7 children suffer from the disease. Secondly, they develop the disease at a very young age. Thirdly, the family is inbred, which tends to be associated with inheritance of disease. And then finally, each of the diabetic children is insulin dependent. So what we can tell from this is that their beta cells either failed to form, they were subsequently destroyed or died, or they just aren't functional. So in order to identify the pathogenic mutation, I first use whole genome sequencing to identify mutations that are homozygous in one of the affected children and heterozygous in both the parents, and then heterozygous in the, homozygous wild type or heterozygous in the unaffected children. So once I've done this, I then use IBD mapping using SNPs to identify regions of the genome that co-segregate with the disease. And then once I have these regions, I can go back to my whole genome sequencing data to identify homozygous mutations within these regions. So to find out what these mutations are doing, I generate iPS from one of the affected children and one of the unaffected children, and then I try to differentiate these into pancreatic progenitors and subsequently beta cells. So here you can see 2 iPS cell lines we derived from the family above. So first thing we'd stain for OCT4 just to check that we have what appears to be a pluripotent line. We'll then do karyotyping analysis to ensure they're genetically stable, and finally we carry out microarrays to ensure their transcriptome is consistent with pluripotency. So as Ray showed you earlier, we're using the STEMDiff pancreatic progenitor kit to turn our iPS firstly into pancreatic progenitors. So Ray showed you the trajectory of gene expression, which is consistent with cells passing through the definitive endoderm gut tube towards the pancreatic lineage. So on the left, so what I really want to know is exactly how far along the pancreatic lineage have they got, first of all, and secondly, can we derive these cells from different genetic backgrounds consistently, which is obviously absolutely essential for disease modeling. So what you can see here on the left is a schematic showing the different proteins that are expressed during pancreatic development, which is from a paper by Rachel Jennings in Neil Hanley's lab, where they stained human fetuses at different stages of development. So at Carnegie stage 12, the pancreatic endoderm has emerged, which expresses PDX1, FOXA2, SOX9, GATA4, and SOX17. As these cells progress towards pancreatic bud cells, they downregulate SOX17 and upregulate GATA6 and NKX6.1. And then upon further differentiation towards the acinar and endocrine and duct lineages, they downregulate GATA6 or a combination of GATA4 and GATA6. So what you can see here are the expression of 4 different genes, so the log2, the expression relative to iPS cells of 4 different genes in definitive endoderm, and then cells differentiated from 4 different cell lines. So in blue we have 2 human ES cell lines, H9 and HES3, and in green 2 iPS cell lines, so a healthy one and a diabetic one. And the take-home message here is really the 4 different cell lines give rise to pancreatic progenitors, which express fairly consistent levels of these key markers of pancreatic development. So looking at SOX17, GATA6 and NKX6.1, we can see they have in fact downregulated SOX17 and upregulated GATA6 and NKX6.1 to fairly consistent levels. And what I take from this is that, what I can conclude is that this protocol is making pancreatic bud cells that are equivalent to the Carnegie stage 13, the cells of the Carnegie stage 13 embryo, and that we were able to do this consistently for several different pluripotent stem cell lines. So given that, what I next wanted to know was how efficient is this differentiation protocol. So here you can see 4 representative FACS plots for the 4 different lines I just showed you, showing the expression of PDX1 and NKX6.1 protein. And so in each case we get a really great efficiency of differentiation. This bar graph just shows you the average of these 3 different cell types across 4 independent differentiation experiments using these 4 different lines. And what you can see is we get an average of about 80% PDX1 NKX6.1 positive cells, and that the variability within the protocol is fairly low. So that's great. We can produce these cells reproducibly and efficiently on different lines. But what I wanted to do next was to find out, can I capture these cells? So this is the endpoint of the stem cells differentiation protocol. So can I capture these progenitor cells and expand and maintain them in culture? So why would I want to do this? Firstly, there's the issue of convenience in that recapitulating the entire developmental pathway, every single experiment, it takes a lot of time, effort and money. So secondly, there's the issue of stability, and that static cell populations enable much more detailed studies of specific developmental states and the transition between them than, say, the heterochronic populations that you get from differentiations that are kind of charging through development. So the first thing I want to do with this system, to exploit this system, is to study the signals and the transcription factors that are required for the maintenance and expansion of the pancreatic progenitors themselves. And the next thing I want to do is to use these cells to undertake high-resolution studies of the developmental cues that are required to generate endocrine cells. So how did I establish these cultures? Well, first and foremost, I take the stem cell technologies differentiation day 15 cells. I then passage these as single cells and plate them on a layer of 3T3 feeder cells. So over the course of about a year, I tried many different combinations of signaling factors and ultimately came up with a system that both drives the expansion of these pancreatic progenitors and also blocks their further differentiation. So these cells can be cultured up to passage 20, enabling approximately 8 to the 20-fold expansion over a period of 20 weeks. So I usually culture them as clumps, just as we culture human ES cells, but they can also be plated at clonal density, which is important for doing things like colony assays, which enable you in the stem cell field to determine the effects of particular signals on both proliferation and differentiation. And I think one of the most important things is that I'm able to freeze and thaw these cells so I can make stocks from many different lines without having to continually go back to the pluripotent cells, which is a real boon for me. Okay, so what exactly are these cells? So I've shown you the same schematic I showed before, listing the proteins expressed at different levels of pancreatic development. And so what you can see here is again the log2 relative expression versus iPS cells in definitive endoderm, again derived from the stem cell technologies kit, and in a human ES, the H9 human ES cell line, and one of my healthy iPS cell lines. And I compared the endpoint of the stem cell technologies differentiation with early and late passage early pancreatic progenitors, or EPP as I now call them. So what you can see from this data is, although there is a small decrease in the levels of PDX1 and SOX9, my EPP cells express these key markers of pancreatic development robustly. They express them similarly between the different lines, and they don't seem to change much over time, as you can see between the early and the late passages. So in addition to these genes, the cells have further downregulated SOX17 and maintained GATA6. However, they seem to have lost NKX6.1 expression, which suggests that these cells have tracked back a little bit and now sit somewhere between the pancreatic endoderm and the pancreatic bug. However, I'm not too worried about the loss of NKX6.1 expression, as I can easily reactivate it when I further differentiate the cells, as I'll show you later on. So just to see how homogeneous my cells are, I carried out a series of immunostaining for the same proteins. So PDX1, SOX9, GATA6 and FOXA2 all expressed very homogeneously, as is the epithelial marker E-cadherin. So to confirm this, I did a FACS experiment which shows 86% PDX1 positive cells and 92% E-cadherin positive. So as I expected, I didn't find any NKX6.1 positive cells, and I found less than 1% of neurogenin 3 positive cells, which indicates that the cells can escape the conditions that I'm using to trap them, but at a very low efficiency, which is what we would expect from any cell line, such as a human ES cell line, for example. So now I know what these cells express and I kind of know what they're made of. The question becomes, what can they do? So I want to know, are they multipotent? Can they form the different pancreatic lineages? And also, can they go on to form functional cells? So the first way in which to test this is to try and put the cells back into a differentiation protocol and push them towards functional beta cells. The second means of doing this is to put the cells under the kidney capsule of an immunodeficient mouse and to see which of the pancreatic lineages they're able to form, so the duct, acinar, or the endocrine lineage, and within that lineage obviously the beta cells and the other hormone expressing cells. So in order to work out how to differentiate these cells towards beta cells, I decided to compare 4 different beta cell differentiation protocols. So Ray has already introduced the first 3 from the Melton lab, the Kiefer lab, and the Hibrock lab, and I also added in this paper from the Deng lab from 2009, as they use a variation of the ViaCyte protocol followed by a brief progenitor expansion step without passaging in this case before trying to further differentiate their cells. So obviously I didn't go back to the start with these protocols. I kind of introduced my cells at the PDX1 positive progenitor stage. And in the first instance, I essentially just looked at the first transition, which in the first instance leads to the upregulation of NCAC 6.1. So to determine which protocol was best able to reactivate NCAC 6.1, I compared each of the 4 protocols side by side using 3 different platforms. So first of all, a monolayer differentiation, secondly, spheres of cells in suspension on a mutator, and finally smaller spheres that we embedded in matrigel. So what you can see here is the gene expression for PDX1 is that gene expression for PDX1 at the end of the first stage of each of these protocols. What you'll notice is that the matrigel condition for the Kiefer and Melton protocols is missing, as the cells in this case mostly died off, so we were unable to get RNA. So in the first instance, none of the different protocols, with the exception of the matrigel ones or platforms, seem to show any real significant differences in PDX1. However, in terms of upregulating NCAC 6.1 expression, the Hebrock protocol, both using the monolayer or suspension platforms, is clearly the best at reactivating this gene in these cells. So in addition to reactivation of NCAC 6.1, I also wanted to ensure that I wasn't prematurely reactivating neurogenin 3, as this will typically result in the formation of polyhormonal immature fetal-like cells, which is obviously something we don't want. And again, the Hebrock protocol doesn't significantly induce this gene. So ultimately I decided to stick with the Hebrock protocol using a suspension platform to further differentiate my cells. There wasn't a lot of difference between the monolayer and suspension protocols at this stage. However, the feeling from the literature seems to be that later on the cells exhibit much higher differentiation efficiencies if they're grown in a 3-dimensional system as opposed to a 2-dimensional one. So this is what I decided to move forward with. So on the left here you can see another schematic I've made, this time of the whole Hebrock protocol, which consists of 3 different stages. So in the first stage, PDX1 positive, SOX9 positive pancreatic progenitors, or EPP, sorry, are treated with a mixture of EGF retinoic acid and FGF7, and this should give rise to pancreatic bud cells that upregulate NCAC 6.1. So in stage 2, I use a combination of PKC activation, ALK inhibition, BMP inhibition, and FGF7 to drive cells towards the endocrine lineage, which is marked by downregulation of SOX9, transient expression of neurogenin 3, and then ultimately activation of NCAC 2.2. Then in the third stage, we don't expose the cells to any growth factors and just kind of develop on their own, and this is supposed to give rise to mature functional beta cells. So what you can see up here is firstly the expression of various different genes in the EPP cells from which the differentiation was started, and at the ends of stages 1, 2, and 3, and then in islets taken from cadavers, essentially, so mature human islets. So as expected, we see retention of PDX1 expression, upregulation of NCAC 6.1, and then downregulation of SOX9. We also see upregulation, very strong upregulation of neurogenin 3 at the end of stage 2, which is great. This hangs around for a little longer than expected until the end of the protocol, essentially, which probably suggests that the cells are not entirely synchronized. We see strong upregulation of NKX 2.2, similar to what we'd see in the islets, which is great, and also of islet 1, but not to the levels we'd expect to see in the islets themselves, although this perhaps just reflects the fact that the latter stages of the protocol aren't 100% efficient. So in terms of hormone expression, we see very strong upregulation of insulin, glucagon, and somatostatin after 3 days, although in the cases of insulin and glucagon, the levels are much lower than in mature islets, but this is something that they commented on in the original paper as well. So just to give you an idea of what the cells look like as they're differentiating, at the end of the first stage, the cells form these rather large sphere-like structures with these ruffled edges, which suggest the cells are undergoing branching morphogenesis. And by this point, you have about 71% of the cells expressing PDX1 and NKX 6.1, which is certainly more than enough to continue with the differentiation. So by the end of stage 2, these structures have condensed somewhat and lost their ruffled edges. The majority of cells have upregulated NKX 2.2, and we see neurogenin 3 in a percentage of the cells. We don't expect to see it in all of the cells, as expression is transient, and we know from the mouse that it's typically not synchronized in different cells. So finally, at the end of stage 3, the spheres themselves have further condensed, and then around 20% of cells have gone on to express a C-peptide, which is a proxy for insulin expression. So this is similar to the efficiency reported in the original paper from the Rock lab, as is the rather widespread in insulin expression levels, which we see here. So finally, I wanted to know whether the C-peptide positive cells we're producing, are they monohormonal, or do they also co-express glucagon, for example, which would suggest that they are immature fetal beta cells. And it seems they do not. In fact, whilst we get a few glucagon expressing cells, we really don't see that many. We seem to be mostly getting insulin positive cells. So what I've shown you so far is that these cells are, the EPP, cultured EPP cells, are capable of endocrine differentiation, and that they can form insulin positive cells at what I consider quite a good efficiency. However, in the coming weeks, I haven't yet looked at whether they're really functional. So in the coming weeks, I'm going to be looking at whether these cells are capable of secreting insulin in response to elevated glucose levels. So as I mentioned earlier, the second way in which I need to test the functionality of these cells is by implanting them under the kidney capsule of an immunodeficient mouse. So this serves really two purposes. Firstly, to show, are the insulin-expressing cells we're generating from these cells, are they truly functional? Can they maintain normal glucose levels in an organism, or in a whole animal rather? And secondly, because we don't have good protocols for the formation of duct and acinar cells or any of these other endocrine lineages, if we want to know how potent these cells really are, then we need to put them in a mouse and see whether they can form these different lineages. So that's exactly what I did. So I transplanted my cells under the kidney capsule of an immunodeficient mouse or mice. I left them around 3 months, and then at the end of this time I took out the kidneys, cryosectioned them, and then stained for trypsin, which is a marker of the acinar or exocrine lineage, cytokeratin 19, which marks duct cells, and then synaptophysin, which marks cells that have entered the endocrine lineage. And then I also stained specifically for insulin and glucagon. So this this image just shows you a H&E staining of the implanted cells. And what you can see here are these very nice duct structures which are characteristic of these implants. So firstly, I stained the implanted tissue for a marker of human mitochondria, just to check that the cells I think are implant really are of human origin. And you can see we see large areas of implanted cells which are kind of dispersed throughout the kidney because they've been there 3 months and they're put in a single cell, so they tend to migrate quite a bit. And as you can see as well, some of these cells also express the endocrine marker synaptophysin. What I was rather surprised and rather happy to find was large areas of C-peptide positive cells. However, I wasn't really, well, so far I haven't been able to find any glucagon positive cells, which suggests that either the cells are heavily biased to C-peptide positives, to the formation of insulin-expressing cells, or simply that I haven't looked hard enough. But that's something I'm going to continue with in the coming weeks. So finally, I've been able to identify a large number of cytokeratin 19 positive duct cells, but so far I haven't been able to find any cells that express trypsin, although previous reports where people have implanted pancreatic progenitors suggest these are form very rarely and are quite hard to find. So again, the search continues. So in conclusion, what I've shown you here is that these cells, the EPP cells that we culture, are capable of forming both the duct and endocrine lineages. And again, in the coming weeks, I'm going to be looking at whether these cells are truly, whether the insulin positive or C-peptide positive cells that form in the mouse are truly functional. So can they maintain normal blood glucose levels when we ablate the mouse beta cells with streptozotocin. So in summary, my project within Ray's lab seeks to study diabetes in order to understand better beta cell development. And of course this goes both ways, that the more we know about beta cell development, the more we'll understand about the failure of these cells in diabetes. So more specifically, my objective is to identify candidate genes involved in inherited beta cell failure and try and find out what's going wrong with these cells in the diabetic families. And I do this by taking iPS cells from the diabetic individuals and differentiating these into early pancreatic progenitors. These cells can be expanded massively in culture. You can grow them up to, well, so far we've grown them to 20 passages, but I assume they can go further. And we can both freeze and recover these cells at very high efficiency. So far I've shown that these cells can differentiate into insulin-positive cells in vitro. What I'm next going to test is whether they're truly functional. So can they secrete insulin in response to glucose? And then finally, I showed you that these cells can differentiate into the pancreatic endocrine and ductal lineages in vivo. Of course, finally, the gold standard test of functionality is really to show that they're capable of maintaining normal blood glucose levels following beta cell ablation with streptozotocin. So ultimately, bringing all this together, I would hope to be able to produce functional beta cells from diabetic individuals and their obviously healthy siblings, and to use to compare these cells to find out which genes and which pathways are important for beta cell development, function, and ultimately diabetes. So with that, I'd like to thank everyone for listening, and thank in particular members of the Dunn lab, so Jenica Tan, Sheena Ong, and Eunice, who did quite a few of the experiments we've seen here. Members, so Mohammed and Karine from Bruno Reversade's lab, who identified the families and carried out some of the genetic analysis. Then the people from the IMB stem cell bank who helped make the iPS lines. And then of course, last but not least, certainly not least, Michael Riedel and Jenna Mokia, who provided the reagents are used to differentiate the cells, as well as really a lot of help and advice along the way. So thanks again for listening.
Thank you so much, Jamie and Ray, for the very engaging presentation. We are pretty close to the hour here, but I think we have a little bit of time for a couple of questions. So as our first question, Jamie, have you done any investigations into the dynamics of intracellular calcium responses of your cells?
We haven't yet, but it's something that's ongoing in the IMB, so it's something we will eventually try. But obviously only once we've shown they can secrete insulin in response to glucose.
So I think that's about all the time we have for questions. Thank you so much for joining us for today's presentation, and an enormous thank you to both Ray and Jamie for providing such a great presentation. Thank you so much and have a great day or evening.